on in your environment. So being able to focus on the task at hand that you're working on, and that can be elicited through the Stroop task, which we'll explain in a little bit. So when we look at kind of what some of that literature says, it's kind of all over the place of terms of how background music has a role in cognitive inhibition. So the purpose of this study, we wanted to analyze the effect that listening to preferred music would have on cognitive inhibition in healthy younger and older adults. We hypothesized that listening to preferred music will result in greater accuracy and decreased reaction times, which would indicate a better performance on the Stroop task compared to no music and unfamiliar music. Now, Ella will take you through more of our study.
All right, so for the participants, for the healthy young adults, we had 30 total between the ages of 18 and 35. The exclusion criteria was the Beck's Depression Inventory, the Mini Mental Status Examination, and the mean age was around 20 with a standard deviation of 1.31. There was 63% female and 37% male, and there was 93% Caucasian for ethnicity and 3.5% mixed and 3.5% African American. And then you can go to the next slide. For the healthy older adults, we had 13 total between the ages of 55 and 85, and the exclusion criteria was the same, with the mean age around 66 with a standard deviation of 5.96. There was 7 female and 6 male. And I believe Emma will talk about the next slide.
Yes, so this is just for those of you who aren't familiar with the Stroop task. It's kind of hard to explain unless you see it, but it's a cognitive task, and it asks you to identify the color of the words you're seeing. So like the color of the ink that's presented on the screen. So here are some examples. When the participants saw the different words, these were the 3 different conditions that they saw throughout each trial. So the first one was neutral. This was kind of just unfamiliar words that they probably wouldn't have seen before. So the example, they would see the word helot, and they would have to identify that that word is written in red. And then there's the congruent condition where the color of the word matches the word itself. So you can see that example. The word red is written in red, and so for that example, they would be pushing the red button, which will show you the setup on the next slide, I believe. And then the last condition was incongruent. So this is where the color of the word does not match the word itself. And even just looking at this, you can see this is the one that makes you think a little bit more. This is the one that elicits that cognitive inhibition that Allison talked about. So this, an example would be the word green written in the color red. You kind of have to filter through that and choose red for that word. You can go on to the next slide, thanks. So for our data collection, and again, this was the same across the healthy young adults and healthy older adults, we asked participants to send us 5 kind of high tempo songs that they preferred or that they enjoyed listening to. They did this before coming in for the data collection, and then we randomly selected one of those songs. So you can see that setup. That was the keyboard. The Stroop task was presented on a computer, and participants just click the button, the corresponding color to the word that they saw. So they did 1 practice session, and then once they were comfortable with that setup, they did 1 condition using the self-reported preferred music, 1 condition with unfamiliar piano music, and then 1 condition with no music at all.
Okay, so going into our data analysis, the main outcomes of our study for both the healthy and older adults were reaction time, and this is in milliseconds, and accuracy, so how many times they selected the correct color of the word. And reaction time was basically just testing, again, how quickly they were able to respond to that stimulus. The statistical analysis that we performed was a 3x2 repeated measures ANOVA.
So these are our results for accuracy. There were no significant differences between music conditions for any of the Stroop conditions. Healthy old adults were more accurate than healthy young adults, and this was actually significant in the neutral Stroop condition. You can go ahead and go to the next slide. These are our results for reaction time. So there were no significant differences between music conditions for any of the Stroop conditions. However, there were significant differences between healthy young and healthy older adults for all three Stroop conditions. And all of these, that older adults were slower to respond to the stimulus. You can go ahead and go to the next slide. So the findings of this study did not support the hypothesis and suggest that listening to preferred background music neither enhances or disrupts cognitive inhibition. We did see age effects, and we did expect this. To our knowledge, results from the study were the first to show that listening to preferred music does not have beneficial effects on cognitive inhibition. And further studies will explore whether preferred music enhances inhibitory control in Parkinson's disease. You can go to the next slide.
Yeah, so just some brief acknowledgements. We really want to thank, firstly, Dr. Stegemuller. She's the research professor that we work under, for giving us the opportunity. And we especially want to give just a huge, huge thank you to Dr. now, Patricia Izbicki. She has been the biggest mentor for us in this whole process and has really taken us through research literally from day one when we were freshmen coming into research labs. So it's just been such a pleasure to work with you, Dr. Izbicki. And we just also want to thank our friends and family for supporting us, and many of them for participating in this research study, and of course the participants to our study for helping us make this happen. So thank you all so much for listening and coming to this presentation. And we'll be taking questions in the chat box at the end if you have any.
All right, great job. Wonderful. So let me go ahead and end the show here. And I will go ahead and jump right into my presentation. All right, can someone let me know if they can see my screen?
We can
Yes.
see it.
Okay, perfect. Sorry, I just wanted to get verbal confirmation. All right, fantastic. And everyone is good to go. All right, great. So in a similar vein, I was really interested in looking at particularly cognitive and motor inhibition, but instead of using music, I wanted to see, are there any differences in these two domains in musicians across the lifespan who continue to practice music? And so I always, for some reason, related, stop inhibition time to MC Hammer stop hammer time. So that was my way to diffuse my nervousness at my actual defense and also today. So hopefully you get a good chuckle out of that. And mainly here, I want to just get a brief background as to what is motor and cognitive inhibition. Give me 1 second here. I'm getting some issues here. I just want to make sure I'm pulling up the correct one so I don't have to bore you with the really long one I gave for defense. Give me 1 second here. Okay. Okay, great. So when we talk about cognitive and motor inhibition. Those are words that, they aren't going to be appearing in everyday life. So the way I like to look at it is when you're getting ready for an important presentation or performance, or you're studying for an important exam, or even when you're driving a car, or in my case, when I'm trying to play the piano. These are two of my distractions, my beautiful mini poodle and my lovely fiance, Andrew. We're all in these activities using cognitive and motor inhibition. So cognitive inhibition, we define as stopping or overriding a mental process with intention, or just being able to filter out everything that's not important around in your environment. With motor inhibition, we are simply defining this as suppression of movement. So for example, when you're driving a car, right, you really want your foot on the pedal to be in one spot, right? You want to have control, you want to be able to accelerate or decelerate, right? And unfortunately, as we age, these two processes, we become worse at doing these in a variety of tasks. And especially as we continue to age and the population continues to become larger, this is going to be something we really have to look at. So by 2050, we're going to have more than 2 billion older adults in the entire world. And we're going to have a lot of challenges with that. There'll be economic burden on families and healthcare for sure, but also we're going to have to deal with decline in abilities to complete what we call activities of daily living and individual activities of daily living. And that's simply just going to the bathroom, putting on your clothes, being able to manage your finances, going out to the store, being able to get food, those sorts of things. So long story short, with this whole slide, with cognitive inhibition and aging and what's happening in the brain during this time, we do see reductions in gray matter, white matter, and biochemical changes. And this is a variety of studies that show this. So that's really what I want you to get from this slide. And we see something very similar with motor inhibition and aging and the brain. We see that there is atrophy or reductions in gray matter, white matter, also reductions in different types of tasks that are associated with this. And so long story short, we know that these decrements in the brain and behaviors that are associated with cognitive and motor inhibition are linked to these individual activities of daily living, or being able to do those simple tasks that I mentioned before, like going to the bathroom, putting on your clothes, getting food, managing finances, all those types of things. So unfortunately, there are limited strategies that improve both cognitive and motor inhibition, and we need to study alternatives. And so what I'm proposing is that music training and music practice might be this alternative strategy that would potentially enhance or at least maintain both cognitive and motor inhibition. So there's plenty of studies out there that show that this is a good option and a doable option, also affordable, definitely much more affordable than going to the hospital for a variety of ailments, and minimal side effects as far as we know right now. And what's interesting is there are a few studies where music training has been looked at from young to older adults. So the study by Hannah Platty et al. And also Strong and Mass, they both showed that older adults with at least 10 years of experience displayed better performance in both nonverbal memory and executive functioning. And so you essentially have cognitive benefits if you have at least 10 years of experience. And it's wonderful because you have these long-term benefits. But what about short-term benefits? What if you are just starting music training in your 60s or your 50s or your 70s? Is there anything that can provide some short-term benefits? And surprisingly and excitingly, yes, there is. So Bugos et al., she showed that putting non-musicians, so musically naive, no training whatsoever before, through a music training intervention. And this was around 16 weeks, so a full semester, had positive results on various areas of cognition. So it really seems that even if you are starting at a much, much later age, and these non-musicians, they were older, healthy older adults, that you can still have benefits in cognition with that. So that's absolutely fascinating. So again, there's another study which was really great that also shows that older musicians perform better on a cognitive inhibition task. It wasn't the Stroop task, it was another task, but it provides the foundation of making the claim that we can use music training to see both long-term and short-term benefits in cognitive inhibition and potentially motor inhibition. So to sum it all up, why do we think music training works and could be potentially beneficial? You have the complexity of the music, of learning the instrument. You have a lot of pleasure. You know, most people that take up an instrument want to learn, maybe not children so much, but if we are older, we are probably going to be doing more things that we like. So we have this pleasure component of music training. You have also the motivational component that if you like something, you're going to keep on practicing it. And then also with the studies that I've talked about before, you do have this component of neuroplasticity. So there are also brain changes that are occurring within those studies. So all of these 4 elements indicate a successful intervention, or in this case, indicate that music training is likely to provide benefits in both of these realms. But, and the whole purpose of this study is there is a severe lack of research that addresses both what is going on behaviorally, so how are participants performing in various tasks, and the brain measures or neurophysiological measures that are associated with that in aging musicians. And so the hope is that the research I'm going to tell you about now that I did for my dissertation is going to provide a foundation to design an evidence-based intervention to target and improve specifically cognitive and motor inhibition in aging. So like I told you before, the goal is to look across the lifespan in terms of behavior and neurophys measures for cognitive inhibition and motor inhibition. And so next I'm going to tell you a little bit about how we did that. So for cognitive inhibition, we use something called the Stroop task, which has been mentioned already before. So you see a word and you have to tell me the color, not the word. And while participants were completing the Stroop task, they were wearing this EEG cap. And so if you know this show, I absolutely recommend you watching it if you don't. But it's called The Big Bang Theory, and I thought this was funny. Of course, this is not how we set up the EEG cap, but this is just for show of more or less what it looks like. So participants were wearing this cap while completing the Stroop task, and that gave us both a measure of behavior and brain activity. And in our actual analysis, we were looking at brain activity in terms of something called the P300. So this is a peak that happens roughly around 300 milliseconds after you see the word. And to measure, now switching to measure motor inhibition, what we did was we had participants complete syncopated finger tapping. So participants were tapping on and off the beat. The syncopated part is off the beat. So essentially a metronome would be going in the background, click, 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 and then a participant would have to tap their finger in between the clicks. So it'd be like click, tap, click, tap, click, tap. And it may seem like a relatively simple task, but it is not. And in addition to that, what I had in terms of brain measures, I went ahead and used TMS. And so this was really challenging for participants because I was essentially stimulating the area of the brain where that's involved in finger movement. So all of my participants, I know I tortured you with this, but I appreciate it. So it was a relatively hard task. And then the stimulation that I used to get my brain measurements for this component was something called transcranial magnetic stimulation. And so this was, if you look at the right-hand corner, is more or less what the device looked like. And I would position the device on the primary motor cortex, specifically the hand area. So again, you would have that hand twitch, and this gives me more or less a measure of what's going on in that motor circuit. And so to particularly measure motor inhibition, the technique I use is something called short interval intracortical inhibition, or SICI. And this essentially is going to give me a readout of the inhibitory part that's going on with the hand twitch. And essentially, there's just 2 pulses. Really, the participant generally can only hear the 2 pulses, but this is only additional information if you're interested in the particular technique. And so to measure that hand twitch, we use something called an electromyographic sensor, EMG, and we positioned it on the FDI, or first dorsal interosseous muscle, so really just on the hand. And what I want you to, if anything, out of all this information for the motor inhibition component, the smaller the hand twitch, the better or the greater the inhibition. So we really wanted to see particularly musicians have that smaller hand twitch, because that would indicate that they had better motor inhibition. All right, so that was just a brief intro to all the techniques, and I'm going to go over my results. So this first experiment is the one that uses Stroop task and the EEG cap. And so I have just a nice graphic of what my hypotheses would be. So I predicted that young musicians would have the greatest cognitive inhibition, followed by young non-musicians, then older musicians, and older non-musicians. So musicians would outperform their age group both in behavior and brain activity, but older musicians would definitely not be better than young non-musicians. So that was my hypothesis for this experiment. And like I said before, we used the Stroop task and paired with the EEG, and participants did get a practice trial, and unfortunately there were a lot of stimuli. So the task in total took about 20 to 30 minutes. So I always told everyone that I know if you are falling asleep on me, because I could see a change in the brain waves on the screen. So that was always a fun thing we were able to at least chat about and actually see differences. But going back to the study here, you know, it was quite a long session, but we were able to get good, clean data. So I thank everyone for being patient that participated in this. And so the inclusion criteria for our young adults, we had our cutoff at 18, or basically we recruited young adults between 18 and 35 years of age, and older adults between 65 and 80. For how we defined a musician, it was someone who was currently practicing any kind of musical instrument except for voice. The only reason we excluded only singers is because there are neurophysiological, or there are brain differences in vocalists compared to instrumental musicians. But otherwise, our definition of musician was very broad. It didn't have to be someone with a degree. It didn't have to be someone that's actually performing live. It just had to be someone who was at least currently practicing a musical instrument. Non-musicians for the young adult groups were had at least less than a year of music experience. For my older adult groups, non-musicians were just participants who were not practicing any sort of musical instrument, but there were some non-musicians or older non-musicians that had previous music experience when they were younger, and it was usually about 40 to 50 years prior. And again, we just made sure that there were no neurological or neuropsychiatric disorders, and also that participants weren't colorblind. And what we ended up with for our data analysis was 22 young musicians, 19 young non-musicians, 20 for older musicians, and 20 older non-musicians. And we didn't really see any differences in terms of cognition, education, income when we did testing between musicians, young musicians and young non-musicians, and older musicians and older non-musicians. So for the Stroop accuracy, just diving into the results here, we didn't see any differences between ages or between musicians and non-musicians. The only differences in age that we saw were in the neutral category. But the one that I want you to really aim your focus on is the incongruent, because that was actually the cognitive inhibition portion of the study, and that's what we're really interested. So we saw no differences here for the incongruent, for accuracy. However, we did see differences in incongruent. So this is, again, the cognitive inhibition portion of the study, both in age and both in musicianship. So older adults were generally slower, which isn't surprising. We also found that in the previous study that undergraduates presented, as well as other literature. But what was really interesting is that musicians, regardless of age, were faster. So they performed better, which leads us to think that they had better cognitive inhibition. And in terms of what we found in the brain, we did see that musicians had a shorter latency over SICI. So if you look at the graphic on the right, this is the middle sensor over the brain. Specifically, healthy older musicians had a shorter latency over PZ. And then the third result, healthy young musicians had a larger amplitude over F3. So all three of these results, this greater amplitude and earlier latency, basically we think it suggests that musicians have better inhibition or were able to inhibit more successfully. So for this first experiment, there were some mixed results with the older musicians and non-musicians in the Stroop task. Mainly, we just didn't see differences in the accuracy, but we did see differences in response time. So mainly the musicians had a reduced response time compared to non-musicians. And one thing that was really interesting that we weren't quite sure, so if I go back to this graph right here, you know, we saw these differences in this particular component, this incongruent condition, which indicates cognitive inhibition. But if you look at the incongruent, so on the left and on the right, you see that the bars really look pretty much the same, that they're going from like the black bar, gray, and then the stripe, they're going from lower to highest. And we were like, that trend is really interesting. And sometimes what's been found is that this is potentially maybe not cognitive inhibition totally, but also an element of processing speed. And what we mean by processing speed is being able to incorporate the information you see quickly. And so what we might think, because we see all of these triangles over congruent, incongruent, and neutral, is that maybe it's not that musicians have better cognitive inhibition, but maybe it's that they have a better processing speed. So they're better able to incorporate and decipher information than non-musicians. Again, this is just a speculation because this was an observatory study, so we don't know for sure. But regardless, we know that, that there are benefits either in processing speed, cognitive inhibition, or probably more likely a mix of both. And so to conclude, for this first experiment, it seems that musicians have enhanced processing speed and maybe a little bit also of enhanced cognitive inhibition in their behavior, and potentially that's reflected in the brain differences that we see. But regardless, this is the first study that has shown these types of results, specifically using EEG and the Stroop task. And it suggests that playing music may be beneficial for older adults and may improve quality of life. So to go on to the second experiment here, this was the motor inhibition component for musicians across the lifespan. And so we also have a similar hypothesis that young musicians are going to have the most enhanced inhibition of all of the groups, and that older musicians will be better than older non-musicians. And so this was sort of a representation of what the experiment looked like. And you can see the picture on the right-hand side. So like I said before, that to get our brain measures, we stimulated the primary motor cortex, or the hand area. And this particular experiment, this was while participants weren't moving. This was just while they were at rest. So we could get a sort of baseline measure of whether there's actually differences even at rest between older musicians, or between musicians and non-musicians, and older adults and young adults. So we had the same inclusion criteria. The only thing with this particular experiment that was very difficult is, as you can see, there are a lot of exclusion criteria. So this was one of the biggest challenges with my study, is that to be able to do the transcranial magnetic stimulation portion, or the TMS, you basically have to be very, very healthy. You can't have any metal in the head, neck, spinal cord, no pacemakers. You can't be on any antibiotics, any antidepressants, any antihistamines. I mean, you typically just have to be perfect, which young adults, it's a lot easier to find, but as we get older, right, life happens, and we have different health issues we're dealing with. So this was something that was a big challenge in the study. But I was still able to recruit a decent amount of participants. So in this case, we had 19 young musicians, 16 young non-musicians, 13 older musicians, and 13 older non-musicians. And same age range, between 18 and 35 for the young, 65 and 80 for the old. And we also here did not see any differences in groups in regards to education, cognitive levels, gender, age, things like that. So here on the right is a representation of where I stimulated on the brain. And long story short, we saw no differences between young and old, and musician and non-musicians, while participants, or while they were at rest, which was really, really surprising to me, because I would have thought at least that I would have been able to see young and older differences, or basically an age effect. And this is both on the left, this is both with an excitatory measure of brain activity, and on the right is that inhibitory measure of brain activity that we are really interested in. And so no differences in either, actually. Then we also calculated, based on some previous research, something called inhibition percent. So this was the amount of inhibition present during our stimulations. And we also saw no significant statistical differences between the groups, even though you can see that it seems like there's a difference between young musicians and young non-musicians, and potentially older musicians, but these were not statistically significant differences. And so, long story short, what we think is going on is that instead of there being what we call cortical mechanisms, so these are the types of, this is like the brain activity that we measure at the surface of the brain, we think that there may be like spinal mechanisms that are potentially involved, because when we were looking at what we call background EMG activity, or basically muscle activity that's going on without the stimulation, with older adults, there was a lot of noise, or a lot of activity going on. And so what's been thought in previous research is that when you have all of this muscle activity going on just at rest, it may be that there's something going on within the spinal cord or spinal mechanism. So potentially the reason why we see these kind of spinal mechanisms that play in older adults is compensation. So that might be the case. We're not totally sure, but it seems like that might be. But overall, it seems like there isn't anything in terms of motor inhibition that's being affected as we age, which is really good. So I think if anything, that would be the main outcome that's exciting out of the study. And long story short, it seems that GABA, this inhibitory neurotransmitter in the brain, seems to be intact both in younger and older adults while at rest. And to go on to the third experiment, this is where we incorporate the actual movement. So we know, okay, it seems that baseline, both younger, older adults, musicians and non-musicians are the same, and there's no issues going on there. But what about when we start moving? What's going to happen there? So the same hypothesis as before, young musicians are going to have the greater levels of inhibition, followed by young non-musicians, older musicians, and older non-musicians. And so what we had our participants do is 85 synchronized finger taps. So they were tapping along with the metronome, or they tried to tap as best they could with the clicks of the metronome, and then 83 syncopated finger taps. So this was between the clicks of the metronome. The metronome was at 1 hertz or 60 beats per minute, so it was wasn't extremely fast, in my opinion. And then while participants were completing these taps, they either got a stimulation that was excitatory, so the single pulse, or inhibitory, so that's the SICI. And we gave 10 stimulations for each session of the finger taps. And they were supposedly given randomly. I can't say for sure whether participants were able to predict when those stimulations were coming, but that was the goal. And here we have the similar demographics to what we had when we did the experiment while participants were resting. So 19 young musicians, 16 young non-musicians, 13 older musicians, and 16 older non-musicians. And the young musicians, again, the young adults between ages of 18 and 35, and older adults between 65 and 80. And again, we saw no significant differences between the groups. So what were our results? So first we'll talk about the finger tapping. So on the left, we have our synchronization, and on the right, we have our syncopation. And so we have, the black bars representing the young musicians, the gray bars representing the young non-musicians, and then the stripes, black stripes, older musicians, gray stripes, older non-musicians. So if we look on the left at the tapping on the beat or the synchronization, we only saw an age effect. So older adults in this case were actually less accurate in synchronized finger tapping. They were about at 39%, and then the young adults were at 56%. But in the syncopation, so the tap that's actually eliciting the motor inhibition, we didn't see any differences whatsoever, which was surprising. So we also looked at what we call the timing or the accuracy difference. So essentially we're looking at, well, how off were participants from tapping where they needed to actually tap? So if we look at tapping on the beat, which is on the left, we did see a main effective age. So older adults generally were tapping 67 milliseconds after, while young adults tapped only 17 milliseconds after. So young adults were they were better at the timing part for the actual tapping on the beat. But when we look at tapping in between beats or the syncopation, we see a group effect, meaning that there were differences between musicians and non-musicians. So musicians were tapping 27 milliseconds after the tone, while non-musicians were tapping on average 27 milliseconds before the tone. And so what we think that this may mean is that there are some differences in motor inhibition in musicians and non-musicians. And I'll talk about a little bit more in depth as to our interpretation for this result on the right in the later slide. All right, so we know behaviorally that there's something happening, but what is happening with the brain activity? And when we look at excitatory brain activity, we see that there are differences, but none of these differences were statistically significant. And that was really interesting. So even though there look like there are differences, it was not a statistically significant difference. And when we look at the inhibitory brain activity for tapping on the beat, which is on the left, again, we see no differences. And when we're tapping in between the beat, we also don't see any differences. And so this was also pretty surprising. And then we looked at this inhibition percent. So we also saw no statistically significant differences between the groups when tapping on the beat. And the same thing for in between the beats, which was also surprising. So there just seems overall that even though there looks like there may be brain differences, they're not enough to be statistically significant. I'll skip over, or actually, no, I won't skip over this one. So we were also interested to see whether or not would age, group, and the inhibitory brain activity going on predict how well someone would perform, regardless, how someone would perform in the finger tapping, specifically the syncopation or the motor inhibition. And we saw that being a musician and also having greater amounts of inhibition actually did predict performance. And this was a different statistical technique we used. We used a stepwise multiple linear regression for this. So there seems to be this predictability, but when we just look at differences between groups, there's nothing there. So it's a little bit strange in how we would interpret what we see. So let's talk about why we didn't see many differences in terms of accuracy as well as brain activity in the finger tapping. So these tasks are relatively simple, the finger tapping tasks. I know some of you that are maybe non-musicians are like, that was not simple, but in retro, in the entire literature, tapping a finger, one finger may not be enough to produce individual or differences in brain activity or behavior. So maybe if we did a bimanual arm and hand movement, so we had both fingers tapping or both fingers tapping on different beats, that would probably produce a larger age effect than just a unimanual task or just tapping that one finger. Also, maybe increasing the beats per minute might be something that would have allowed us to see differences. So usually what we've seen in the literature is that the higher movement rates are less stable in older adults. And also what might be an issue is repetitive time finger tapping tasks have similar accuracies in young and old adults. And that's exactly what we did here. And so there's other literature that backs this third bullet point up, that if you're doing something that's extremely repetitive and timed, it really doesn't make a difference if you're younger and older. So all in all, the reason we might not see differences both in age and group for the finger tapping accuracy, it may just be that the task is way too easy. So when we talk about the actual tapping and not just the accuracy of the tap, timing has generally been found to be more accurate in young musicians when we're talking about finger tapping, and that makes sense. We've been taught to tap and be within the time, and especially if you're playing in a band, you better be in the same area and timing as your other fellow musicians and the conductor. And so long story short, it seems that musicians are anticipating better than non-musicians if we look at this third bullet point, and non-musicians seem to just be reacting to the tone. So long story short, this anticipation is potentially leading to better motor inhibition in terms of the timing of the finger tap. And we talked about the spinal mechanisms in terms of the brain activity in our previous experiment, and it seems the same thing is happening potentially here. We have some other data that also showed that while participants were at rest and also at movement, while they were moving, the EMG activity was fairly messy, regardless of the actual collection of the stimulation or the brain activity. So also we were measuring brain activity after the movement occurred. And so the pathway actually responsible for the movement just may simply not be activated. So it might be also be that's why we didn't see any differences. And the third bullet point, the somatosensory component is crucial. And what I mean by that is those of you that participated, when you were finger tapping, you were just finger tapping in the air. You weren't hitting any kind of tabletop or any sensory or surface. And when we're playing a musical instrument, that is essentially what we're doing, right? We're making that somatosensory or that physical connection with our instrument. And if you take that away, maybe you're just, that's why we don't see those differences. You're taking that somatosensory component away from the musicians, and it just doesn't matter whether you're a musician or non-musician. So if we actually had, we allowed for participants to tap on a surface, then we would have actually seen a difference in terms of brain activity. So long story short, we didn't see that there was a direct enhancement of motor inhibition in musicians because we didn't see those differences between the groups. Might be because of a simple task. And it's great because it seems that it doesn't matter across the lifespan, we are still able to do simple tasks like this and there's no differences. However, when we did that other statistical analysis, that multiple linear regression, it seems that musicianship and the levels of inhibition do predict how well you do on tapping between the beats. So it indicates that there is something more complicated going on there in terms of musicianship influencing motor inhibition throughout our lives. And so long story short, I think there is some evidence here to say that music practice could maintain motor inhibition or improve it across the lifespan, but we would have to do more studies with more complicated tasks to be able to see that both behaviorally and in the brain. So the last experiment here, and I know we're running short on time, here this was mainly an exploratory analysis to see whether or not we saw any predictions in terms of musicianship and brain activity for all of our tasks. So basically, does musicianship and brain activity predict how well you're going to do in both the Stroop task and the syncopated finger tapping task or tapping in between the beats? So we predicted, long story short, that there would be, that musicianship and brain activity, inhibitory brain activity, could predict better performance. Unfortunately, with our results, we actually didn't find that, which was very interesting. And let's see here. But the caveat is that we didn't find that the brain activity was a significant predictor of performance. We only found that age and musicianship were. So the younger you were, the younger you are, and if you're a musician, you would have better performance. So that's why there's this X and check mark here. So the third bullet point just said that. And then number 2, we thought that also for the motor inhibition, that greater inhibition and being a musician would predict what we found. So hold on, let me get to this one here. And so long story short, we did see that it seems that at least here with predicting motor inhibition, musicianship, it seems that there is some kind of predictability in terms of what's going on in the brain with cognitive inhibition. And again, this second hypothesis is looking at brain motor inhibition, whether or not that and musicianship can predict cognitive inhibition brain activity. And maybe, sort of, we only found that over PZ, and specifically it was with the latency or how quickly we saw that signal. So long story short, it seems that neural circuits, both in the motor cognitive realm and between musicians and non-musicians, they may be more refined and less overlapping, meaning that musicians may have more refined and less overlapping neural circuits based on the analyses that we ran. And again, I didn't show everything just for sake of time, but I'm happy to discuss more with you. And our task may be activating non-overlapping neurocircuits, separate ones. But again, this was just an exploratory analysis, and so we'll see what will happen once we actually do an experiment on that. So the Big Kahuna conclusion from my dissertation, and the big takeaway that I want you to get from this entire presentation is, in the first experiment, we found that it seems that musicians have enhanced processing speed alongside cognitive inhibition. So both are actually being affected potentially. In experiment 2, we saw that there is no differences in the functional motor inhibition circuitry, so no differences between young, old, and musicians and non-musicians while at rest. With the third experiment, where we incorporated the movement, it seems that music practice and motor inhibition do predict how well you do in terms of performance on the finger syncopation task. However, we didn't see any differences. So this is potentially really the only thing that we can say out of that entire experiment, and would have to be followed up with other studies. And the last experiment, this was just an exploratory analysis. We think that based off the statistical analysis or the multiple linear regressions, that maybe musicians have more refined than overlapping cognitive and motor pathways. But again, this was exploratory, and we would have to follow that up with different studies. So to conclude, I want to thank everyone who has helped me in this process. And I am grateful to my dissertation committee for being flexible during the pandemic and being willing to meet both in person and online. When I defended officially, this was in March, so we still had social distancing, but we still met. 3 of us met in person and 2 of us online. Everyone in the NeuroPhys lab, which is the picture on the right, I'm so grateful to everyone who has helped. Obviously, first and foremost, my fiance, Andrew, who has been just awesome and supporting me, and he's actually defending in a few weeks. So we've both been very busy this semester. All of my undergraduate assistants and my PI, Dr. Stegemuller, who's pictured on the left, just big thanks to all of them. The neuroscience program for supporting me, all of the participants that completed my study. I'm just so grateful, because I know that it was, it was quite the commitment of time. So I'm just so grateful that I was able to experiment on you, and you were my guinea pigs. And then all of my family and friends throughout the years. You've heard me rant and vent and rant again. So I'm just thankful that you were there for me for all this time and just kept pushing me to complete this and go forward. And also my funding sources, I was lucky that I was able to get some small scholarships. The first was through Iowa State, through College of Human Sciences, and then the second was the Iowa Women of Innovation Award. That was actually a big chunk of where my money came from to complete this project. So I'm really grateful, especially to Dr. Sandra Ashmore, who was just so important in getting that award. And with that, I guess we'll take questions for both the first and second presentation, and go ahead, you can type them in the chat box here. Or if you're having trouble with the chat box, you can go ahead and just unmute yourself and tune in and tell me your question, or tell the first group your question.
She has a nice screen.
I'm getting out of here. Oh, thank you.
Yeah.
Okay, perfect. So I guess I'll start with a few questions here. So first one,
Ravenscraft. Must be
trying to
typing.
yes, I'm trying to find whoever is typing and mute them. Oh, sorry, see, I'll mute you. All right, so the first question, is there a possibility of conducting longitudinal studies similar to these with younger adults? So there, I don't know if in our lab that will be possible. There's definitely, it can be done. It's just there, it takes a lot of time, money, and resources. If you're interested in learning more, there is a researcher, her name is Dr. Asal Habibi, and she's at University of Southern California, and I'll type her name in the chat box. And she's actually working with a youth symphony orchestra there and doing a randomized control trial to see whether or not there are actual differences in younger children. So these are all the way from 6 to teenagers, and she's been following them now for 7 to 8 years. So that would be someone that I would recommend looking to for research, for more research on that. All right. So the second question is, do you and your committee believe the study was sufficiently inclusive, or do you anticipate continuing this work with larger samples? So we did what's called a power analysis, which basically lets us figure out how many participants we need to be able to see an actual experimental difference or not. So we believe, based on how we calculated that it is sufficient. But of course, the larger the sample, the better. So that was the way the committee and I approached that question. And this question is from Mary. She asked, do you plan to continue this area of research along the lines you have suggested? It depends. Andrew and I will both be staying for a postdoc here at Iowa State. Our main duties with that will be, to assist in a 29-site clinical trial, and we're one of the sites here at Iowa State with exercise and persons with Parkinson's disease. But we will do side projects. I'm not sure how I will incorporate this area of research. I definitely still want to study musicians across the lifespan. I'm just not quite sure how I'm going to do that yet. So that's a great question. I just don't have a clear answer for you yet. And then Larry, I'm glad that you'll continue playing music for as long as you can, and I would suggest that to everyone. And this next question is from Sue. You mentioned how very healthy your older subjects are. Could they be non-representative, and would that possibly affect your results? Absolutely, absolutely, no doubt in my mind. So that's the trade-off, is that we can, specifically for the transcranial magnetic stimulation, or the TMS, it was unusually healthy older adults. So absolutely, I would not say this is representative of the entire population. And also, I think that the fact that we live in a college town and there's so many professors and people with a higher education, the reason I didn't see a difference in education between older adults and older musicians and older non-musicians was everyone pretty much had a master's and above, and I definitely don't think that's representative of the population. So again, we can just make small inferences, but I would say that for sure not representative of the entire population. All right, is there any other questions that I can address at this time? And feel free to hop on the mic if you want. All right, great. Well, I have recorded this session. If you would like a copy, feel free to email me. And I am just so thankful that everyone showed up. I really think I would have gotten less people to show up at my actual presentation. So it's It's really exciting, and thank you all for supporting me. And I will also keep everyone up to date via email of any publications that come out of this. And I'm just, again, very grateful and thank you, and just have a wonderful rest of your week.
Congrats, Patricia.
Congrats! I love it.
Andrew.
No, that was Tony, I think. I just saw the back of Andrew's head, like from Brandon's screen. I'm like, oh, that's great.
Congratulations, Patricia.
Thank you, Kathy. I'm glad.
Tim.
Whew, that was a lot. It's so bizarre to do this.
Good job.
Thank you. It's so weird doing it online because I get no social cues, and I'm like, all right.
Yeah. But you did great. So you're done now. One more later today.
Yes, one more later today, and then I'm done.
You go at 3 or later.
Hold on, let me stop recording.